uh, lecture 9 of this Gyan series. Um, to recap what we discussed in the previous class, previous lecture, I spoke about spectral domain OCT, different types of OCT, which includes time domain OCT, spectral domain OCT, and snap source OCT. The basic idea is that you have a broadband source which emits a range of wavelengths and it goes to an interferometer in spectral domain OCT and subsource OCT. There is a mirror which in time domain which is moving. You fix the mirror position and you have an array of detectors in the case of spectral domain OCT, which splits the light into different wavelengths, and you have an array as what's called a spectrometer, which measures different wavelengths of light, and then you take an inverse Fourier transform to get the depth axial scan. And swept source OCT works using the same ideas. Instead of having an array of detectors, you have a single detector, but each wavelength is emitted one after the other after the other. So basically, you synchronize, send a wavelength out, make a measurement, send a second wavelength, make a measurement. So it's time multiplexing as opposed to spatial multiplexing. And the same principle is true. You get the detector signal, you get it as a function of frequency, and then you take an inverse Fourier transform and you get your depth signal. That's essentially the idea behind both swept source and spectral domain OCT. And so we will, and in the second half of like in most of yesterday's lecture, I talked about a lot of different applications of different ways in which you can use OCT for biomedical and clinical applications. So yesterday I gave you an example of this endoscopic probe where it looks to the side. It's a pill which you swallow, for example. It looks to the side and it does a circular scan of the side of the entire cross section of the esophagus, and as it goes along, you can get cross sectional images from the mouth to the stomach in the esophagus, the food pipe of the esophagus. So it's a side viewing architecture. So for several organs where it's a tubular structure, it's like cylindrical structure, then you need to look to the side and do a rotational scan and move. So it will be a helical scan of the organ. So that's useful for some organ. Another kind of architecture, another kind of OCT probe that is very useful, for example, for cervical cancer and other some applications, is what's called a forward viewing probe. So instead of the light going to the side and then you rotating it to create a circular scan, you look forward in a forward direction and do an XY scan of what you're looking in the forward direction. And the crucial thing about OCT compared to a regular camera is that you get 3D information. You don't just get information from the surface, but you get depth information. So you get volumetric 3D information from different organs. So this is a paper which we published last year, and from my lab. So this is the essential architecture of what like me and my students built. So it's looks something of like the internal structure looks something like this, and once it's built and made in the lab, it looks like something fairly simple. Something it looks like a thin pen. This diameter is about this is 3 millimeter, 4 millimeter. So it's a very thin kind of pencil, thinner than a regular pencil. So it's something you can hold in your hand that size. Okay, you can put the ruler is on over here. And so the idea here is it uses a swept source architecture. It's swept source OCT. Laser sends light. It's all fiber optic. There is a 50-50 beam splitter, and it goes. One goes to this probe here, and the other goes to a reference arm. And you make a measurement. Light comes back, and there is a detector, laser and detector together, and basically sorry, the beam splitter is over here, and this is for the return data. Sorry about that. Yeah. So the beam splitter is on this side. I apologize for the yeah. So this is the sample arm is basically where the tissue is. Sample arm is where light goes out and you get information. And say, for example, if you want to identify cervical cancer, this like light comes out of this direction. In the previous versions, light came out this way and you do a circular scan around it. But in this new version, you have light coming out this way. And the question then is, how do you do the scanning in the forward direction? How do you, okay, so this, as you know, OCT, all these versions of OCT are point scanning. Sorry for that. Yeah. Okay, yeah. So all these versions of OCT are yeah, point scanning, right? You may measure a point. At one point, you measure the depth. Then next point, next point, next point. So the question is, how do you scan? In the like, if you have it on a benchtop system, then you can use what's called a galvo scanner. You can take a mirror, scan the mirror left to right or up to down, and that way you can do a XY, what's called a raster scanning. Like you can scan horizontally, then come back, scan next, scan next. So that's called a raster scan. That's one way to kind of do a scan. It's like a zigzag kind of scan. But if you don't have, you can't put, imagine if it's something is like 3 millimeters or 4 millimeters, it's so small. You can't put any complicated stuff to move inside. So there are no components which are easily available which can steer the beam. So what one of the things that we did was use what's called a piezo architecture. So how many of you have heard of the piezoelectric effect? Yeah, what exactly is it? Yes, yes, it's that's right. So there are what you said is right. If you put pressure, the electricity is produced, or equivalently, if you put electricity, pressure is there's a deflection. Okay, so both are there is a kind of similar. So one can be used for sensing, other can be used for actually moving things. So in this case, we use the second version, which is you apply an electric voltage, and that will result in a deflection. Okay, so this is a thin tube, and imagine a cylindrical tube of this sort. Top and bottom there are two electrodes. You apply a voltage difference across this way, then this will deflect. The tube will deflect in that direction. Apply there is another two like electrodes here. You apply a voltage here, it will deflect. Okay, so that's the so you have x-axis and y-axis both axis control. Applying a voltage, and the voltage can so there are wires here, which so the wires which are soldered onto the piezo go through this tube all the way to the outside, and like we have a function generator amplifier and things on the outside for piezo controller, which depending on the voltage it will deflect. And the deflection, the thing about the piezoelectric effect is that it's quite small, meaning the like even if it's a substantial voltage, the deflection is very small, like microns, like small amount of deflection. That is not you want to deflect the beam by millimeters, right? Like you want it to be big, like sort of big area, or you want to image a large area. But this deflection is very small. So what do you do? One of the solutions is you can use the cantilever effect, meaning so basically if you make a small deflection, if you have a long kind of fiber here, it will move like this moves a little bit, this moves a lot, right? So there is like a so you can use some kind of deflection mechanism which amplifies your deflection. This imagine this is fixed, this is moving, this moves a little bit, this moves a lot more, right? So there is an amplification to some extent, but that's still small. So what we do is what's called resonant amplification, meaning if you apply this, if you like, so depending on the length of this cantilever and what frequency of or like of the voltage you apply, so this keeps moving up and down, up and down. If that is at resonance with this the resonant frequency of the fiber, then it will start amplifying, like become oscillating a lot. So it results in a large amplification. So you can even move, even if this is moves a little bit, this other end, because of resonance, gets amplified to some like sort of millimeter scales. So this reflects by a millimeter, like millimeter size distances. How many of you have heard the concept of resonance? Yeah, so some of you. Yeah. So like basically, even if a single amplifier, like sort of at just the right frequency for many systems, if you apply the correct, like in this case, a voltage, slowly the mechanical, like the amplification builds up, and it will like small deflection every time it gets in, like it keeps increasing and increasing until it reaches a steady state where it's like a very high amplification. That's the essential idea behind this approach. So this way we can do a big area scan, relative to this size, a big area scanning with a piezo which moves just a little bit. So that's one of the key things that makes this basic this device work. So you apply a voltage. So if you just remember physics, like basic physics, the frequency of, like there is a way to calculate depending on the length, depending on the mass, and so on. There is a way to calculate the resonant frequency. Okay, so now what happens is that if you apply voltage along y-axis, there'll be a deflection here. If you apply the same voltage along x-axis, it's the same frequency. It's like it's initially we made it circularly symmetric, the fiber. So because it's like it's just a single fiber, right? So it's circularly symmetric. So x and y frequencies the same. So what happened was in practice. So even if when you apply voltage along y-axis, it will move along y, but there's like a small kind of deflection along x too. So instead of it being exactly like sort of up and down this way, like when you apply a voltage along, say this is the x-axis and this is the y-axis. So you apply a voltage along y. Ideally, you want it to be a perfect straight line of resonance, but because of like coupling between x and y, you'll see something like this. Like kind of the motion would be like this, and along x, same thing. If you want perfectly like you want it to be like this, but it in practice it looks something like sort of it will be like oval, a thin oval kind of a shape. So this was a practical problem we were trying to resolve, and one of the ideas that we came up with was, so we you want to make something different between break the symmetry. You want to make something different between the y-axis and the x-axis. And the way we did it was like something fairly simple. You take a optical fiber, just cut it into a small piece, like less than a centimeter or so. Glue it, put glue and glue it along the y-axis. So now the resonant frequency, because there is, if you move along, the stiffness along the y-axis is different from the stiffness along the x-axis. And therefore the resonant frequency along the y-axis will become different from the resonant frequency along the x-axis. So this way, if you apply voltage, the resonance will be only along y-axis, therefore it will be a perfect straight line. Then if you apply the different voltage along the y-axis, then because it will be resonant at a different frequency, it will create a perfect straight line. So basically, this way you can completely isolate and make x-axis and y-axis independent, okay, so that you can control exactly what position you want, like, or what shape you want the beam to, like, beam shape to be. Okay, if you want to scan it in a specific manner, you can choose this now. Choose a pattern. I'll get to what patterns you can choose in a minute, but basically that's the idea. And so this way we created a device which everything is, as you can imagine, very small. So to give you a sense of how small these things are, the piezo is about 1 millimeter in diameter. That's the size, outer diameter. The fiber itself is about 200 microns. Basically the width of your hair. like that's the thickness of the fiber. So yeah, so this is about 3 millimeters, this diameter. If you include the cap, it's about 4 millimeters. And we have a lens in front. So light comes out of here, and then there's a lens in front, focuses to a point. When you move the fiber, it focuses to this point, this point, this point. So you can create a focus beam at any position you want, and then keep moving the beam, and you can create a scan. That's essentially the idea. Any questions? Okay. So here is kind of, yeah, the so y-axis we put this like small piece of fiber so that you have a differential, like you can create a control, or like you can control this entire optical system better. And then so we measured the frequency to be 162 hertz for one axis, the x-axis, and 184 hertz for the y-axis. So as you can imagine, if it's resonance, it's a sinusoidal oscillation, right? It's not a linear straight line. So it's sinusoidal pattern along x-axis, sinusoidal pattern along y-axis. So if so, this is actually, if you open the tube inside, this is what it looks like after making it. So this is the piezo tube. So yeah, I'm sorry, it's 1.5 millimeters in diameter, not one. The internal diameter, I believe, is one. So, and then so you this, like I'm showing you the distances. It is about 1 centimeter or so in the fiber fragment, and yeah. So this light comes out of this at this end, and once when it's in resonance, you can see it looks a bit blurred, right? So it's actually resonant along this axis. So like although this side looks like it's fixed almost, this when it's in resonance, this like beam, like uh, the light coming out of here can be much like sort of it's fairly substantial deflection. So when it's in resonance, you can see that. Second thing is, here, if when you apply a horizontal voltage, like voltage along the horizontal, you can get a perfectly straight line. If you apply voltage along the vertical, you get a perfectly straight vertical line. So it means x and y axis are completely independent. There is no coupling between them. So it works quite well in that way. And the next thing is, what kind of scanning pattern should you use? So if you apply a sinusoidal voltage of one frequency along x-axis and the sinusoid along y-axis, turns out that you get these kind of patterns. Have you seen this before? Lissajous. These are called Lissajous patterns. You might have done this in your physics lab or some other kind of previous, you might have seen this in a few other places. So basically, so this kind of pattern is extremely useful because it covers a large area. So if you imagine a raster scan is basically one after the other kind of a thing. So it's a linear kind of a scan, and then so after the end of this one, you go back to the beginning. After the end of this one, you go back to the beginning, and so on. That's the nature of a typical. This is the typical scanning mechanism people use. For if you have enough control, if you have mirrors, you can do this kind of scanning. But in our case, it's sinusoidal. If you have a sinusoid along x-axis, sinusoid along y, and it's actually harder than you think because you have to have a like perfect, like they have to be integer multiples of each other for it. This is the Lissajous pattern to be stable. But we achieved that through some waveform generation mechanisms, and you can create a pattern of this sort. Another big advantage of this Lissajous pattern is that as you keep increasing, you can choose the frequency difference and you can choose different patterns. So you can see that this entire thing has, like, you're fully scanned. Like once like it goes a certain amount of time, it would fully scan and fully cover the entire field of view. It covers the full area. But somewhere in between, it looks like this, where there are gaps in here. So like so. Can everyone see this on the? So the patterns are here, and then there are kind of gaps here. So what this means is that like instead of say if you had a regular raster scan, after like let's say the entire scan takes 1 second, after like 0.3 seconds it will cover only a small area, zero point 0.4 seconds and 0.5 seconds and so on. So every like it will cover it linearly. But in our case we have the big advantage that we don't need to do any of the kind of linear versions of scanning. What we do is uh, um, kind of so you go this way and this way, so on. So it I'm not drawing it well, but it's a nice clean pattern. You cover the entire area very quickly and then repeat and fill in the gaps as you go along. You fill in more and more of the gaps as you go along. So the big advantage of doing this way is that you can actually stop somewhere in between, and you can like if you can kind of if you are okay with there being some gaps, or equivalently if there are some other methods where you can fill in the gaps, then you can do imaging much faster. You don't need to wait for the full image to be collected to kind of finish the kind of get all the data. What do I mean by that? So let's say there are some gaps here. There are some algorithms like computational algorithms called in the field of what's called compressive sensing, and where you might not have known about this, but the essence of this idea is that even with limited amount of data, you can fully construct a full image. Even if there are gaps in the image, like you can fill in the gaps perfectly under some constraints of, they call it sparsity. Like if the data is sparse in some particular domain, then you can fill in the gaps perfectly. And if you, there people have shown this mathematically. This is not like kind of machine learning type kind of thing. It's actually under some specific mathematical constraints. This is guaranteed to work all the time. Kind of, if the conditions are true, it will always do a perfect job of filling the gaps. Okay, so basically with that computational approach, we collect less data and fill in the gaps and get full reconstruction. So here is kind of here are the results of such a reconstruction. So if you take 0.2 seconds, like you can't, it's not enough. There are a lot of gaps, and so even with 0.2 seconds, actually, you can see what the reconstruction. So there are a few different algorithms. There are two of these algorithms which like compressed sensing algorithms which we use. One of worked like didn't work well. The other one, it's a bit blurry, but like in the end, the image looks something like this. The ultimate like image that was trying to see looks like this 10 and these lines on it. Okay, so, but even with like small, instead of this took 1.83 seconds to collect the full data, but with just 0.2 seconds, which is so much shorter, you can still get like very close, right? Like sort of you can already see that like with fill in the gaps, you can see that it's getting close. And with 1.3 seconds, it's almost near, like almost perfect. So instead of 1.8 seconds, 1.3 seconds is like a fairly substantial 30 to 40% speed improvement by using this kind of a combination of this such scanning with computational approaches. We were able to show and quantify that this like this approach to scanning, and this, so the device that we built, the fact that it scans in this particular manner, using that scan to improve the speed of data collection using some computational approaches. A combination of all these things makes this device quite useful. And the one of the things that makes this different from other OCT methods is it's forward scanning, and you can scan in the forward direction. Doctor can hold his pen and keep looking, and they can get a 3D image of what is in front of the device. Okay, that's the essential idea. So this can be used for say cervical cancer or that several of these kind of problems. So this was published last year, and you can take a look from this from my lab. Any questions? So this wraps up my discussion of OCT for this lecture. And next big topic I'll talk about is spectroscopy. And that's the rest of today's lecture. So before I move on, any questions about this or OCT in general or anything else to discuss? So that's discrete wavelet and discrete, so the discrete wavelet transform and discrete cosine transform. So those are some of the details of, this compress sensing, the idea, the method that I mentioned about computation method, requires that the data is, after doing some transformation like the Fourier transform, wavelet transform, cosine transform, after doing some transformation, there are most of the data is zero. Only there are only few non-zero components. That's called sparsity. Okay, so after what transformation will there be the smallest number of like the coefficients? So in terms of like using DCT, discrete cosine transform, is a good transform for the purpose of this like data processing. So we tried several different transforms. We tried different approaches. These are some of the better ones which we found. Yeah. Yes. Uh huh. Sure. I'll this one or this one. Okay. Go back. Oh yeah. Okay. Ah yeah. I see what you mean. Yeah, yeah, yeah. So I see what you mean. Yeah. Right. So the problem is that it's inconsistent. Like the issue is not that it is like as long as you can know the shape for sure, you apply a certain voltage. Sometimes it's a straight line. Sometimes if there is a small error, right, it will become an oval. Like so, it becomes like unpredictable. The problem is not that it's oval shape. If we can reproduce usually 100% of the time, it's always the same shape, then we can use it and make use of it. But the problem is sometimes it's a straight line, sometimes it's a big small oval, sometimes it's a cycle because so if there is cross coupling between x and y, then it becomes like a bit unstable. Like it's hard to kind of make it do the same thing repeatedly. That is the real problem. Like yeah, that's why we wanted to make it like perfectly horizontal and perfectly vertical. When you apply zero voltage along y-axis, you want it to be x and nothing else. That gives you full precise control, and the control is really the crucial piece. Uh, as long as it's predictable, it's okay. And this is like straight line is a very predictable thing. That's a good question. Any other questions? Any questions on that? Yeah. Mm-hmm. That's a good question. So you can see, so we have attached this over here. So the cartoon is probably easier to visualize. So we attach this kind of fiber here along the y-axis. This, so imagine if this has to move up and down, right? The stiffness of this along the y-axis would increase because you have put something more rigid. So the stiffness along y-axis. So if you instead of having say one kind of stick, if you have two sticks on one on top of the other, and you move it up and down, it has more and it's harder to move it up and down, right? So in some sense, its stiffness has changed. But if you like move it in the other direction, it's only just one stick, and therefore the stiffness along y is different from stiffness along x, and the stiffness is essentially what gives you the resonance is related to the resonance. Does that answer the question? Any other questions? Okay. All right. So with that, I want to move on to the next big topic I wanted to cover in this lecture series, which is spectroscopy, specifically absorption spectroscopy. And to recap something which we discussed last week, so when we talked about light matter interaction, there are all these different phenomena, the processes that can happen, right? It's like a wave is incident, it can get reflected, it can get diffracted, it can get refracted, it can get scattered, it can get absorbed. So there are all the different imaging modalities that we come up with. Each of them uses one or a combination of these phenomena. And what um, the like the what we will discuss today is some devices that use absorption as the mechanism for contrast. And specifically, not just absorption at one wavelength, but a range of wavelengths. So the basic idea of spectroscopy is that you make a measurement, say, of absorption at one wavelength, another wavelength, make a plot of this absorption, for example, and that there is a variation, there is a specific pattern to the absorption. Say, for example, some wavelengths may be selectively absorbed, some may not be absorbed, and by looking at this, connecting the pattern of absorption to what is the molecular composition, you can figure out what is, say, atomic composition, molecular composition. You can figure out what is going on inside by making measurements, spectroscopic measurement, meaning the measurements as a function of wavelength or wave number. That's the essential idea. So here, for example, uh, uh, this is the visible range, this is the ultraviolet, this is the infrared. So there is the, like, say, water absorbs, has a certain absorption of range. And for example, here this is oxygenated and deoxygenated hemoglobin. I mentioned this once before. So basically, this is used in what's called pulse oximetry. If you go to a hospital and if somebody, they put a clip on you, and if you're at the bedside, and they keep beeping about once every second or so, right? Basically, that beeping is because of this pulse oximeter. It looks at the blood oxygen, like, at different wavelengths. If blood has good enough oxygen, it will have one profile. If it does not have enough oxygen, it will have a different profile. So making measurements at two different wavelengths of light, this device, that clip that you put, it can figure out how much so in fact, like, first of all, it will figure out how many beats per second, like how much what is your heart rate and how much blood is flowing. It can also tell you what is the blood oxygen saturation, how much oxygen is there in your blood, which can be crucial. If it goes too low, then the person needs immediate medical attention. The doctors and nurses will run towards the patient, so that, like, if it takes too long, then the person could die. So it's crucial. That's why they keep put it on the bedside and kind of use that as a that's one of the devices that is built based on spectroscopy. Okay, so light is when you're using light, you can use light of different wavelengths to make measurements. My interest, one of the things that I do in my research, is what's called mid-infrared spectroscopy. And why mid-infrared? So if you think about how different atoms and molecules, like how they interact with light, so you might have seen in basic physics that if light of certain wavelength or frequency is incident on an atom, then there are electronic structures, electronic levels. Electron can go from low level to a high level, and then the light gets absorbed. And if it comes from the high to the low level, it emits light. Those are called electronic transitions. Electrons move up and come back. And so those happen in the visible, typically some in the UV, but typically in the visible range of the electromagnetic spectrum. Visible light corresponds to electronic transitions. But there are other kinds of transitions. For example, if you take a CH, like, bond, carbon hydrogen hydrogen bond, carbon hydrogen, like this, can be kind of vibrating. Like, so a certain frequency of light can come in and it can absorb, like, if that is absorbed, it can induce this vibration of certain sort, or it could be anti-symmetric, like, sort of these kind of vibration of these CH bonds is symmetric, like in different kind of orientation. This absorption, these are called vibrational levels. Just like you have electronic levels for of transmission, you have vibrational levels. These are quantum mechanical levels. And so, if there is one level of, for a molecule at a certain position, and there is a second level, if light of that difference in frequency, difference in energy comes and is incident, that light will get absorbed, and the molecule will transition from one level to the other level. That's the essential idea here. And the reason this is useful is uh, if you have, so one set of bonds has a certain, like one or few peaks at which it absorbs very specific wavelengths of light. Then if you have, instead of CH, you have CO, carbon, oxygen, and like nitrogen, hydrogen or carbon, nitrogen. So there are different molecular bonds, right? So any molecule consists of a combination of molecular bonds. And if you know the combination of molecular bonds that are present, then for many of the organic molecules, you can figure by making a spectral measurement. So you make a measurement at a large range of wavelengths. Each of them has a peak at a different location. Then you can, by taking the combination of these molecular bonds, you can figure out what molecule it is. Okay, so this is called vibrational spectroscopy, and this is actually one of the kind of more important ways and one of the best ways to identify molecules. Organic molecules in particular is a very good way of identifying chemical composition. Say, for example, a forensic person, goes to a crime scene and they find some unknown chemical of some sort. They can bring it to a lab and put it under this vibrational spectroscopy instrument and make a spectral measurement, and then there are databases already of like about, so there different, like people have developed these databases of 125 150,000 chemicals. Each of their spectra has been measured, and in fact a society that I am involved in called the Koblenz Society actually initially did this work and has a compilation of all these spectral databases. And you do use some algorithms to database matching. The unknown spectrum, you can match it to this library of database of spectra. That way you can figure out, oh, this is this chemical or this is this molecule, that kind of thing. So you can identify unknown chemicals. It's often used in chemistry labs as well to do this kind of. So if it is a single chemical, it's like, as in, people have been doing this for a long time. But if you have a mixture of chemicals, that's when it gets a bit more difficult. But the basic premise is like, so first I'll talk about the simplest version, which is if you have a single version, what happens is, if you have light incident of a certain amplitude and a certain wavelength or 3.5 microns, or so in this lecture, lambda, which is the wavelength, is 3.5 microns. Wave number, which is 1 over lambda. Okay, that is the common, like, that's because of historic reasons. If you use wave number here, it is 28. In this case, 2853 is the wave number. So people talk in wave numbers, frequency in this field of spectroscopy. And so light of this frequency is incident. It gets absorbed. So some light comes in, some light goes out. The amplitude of light going out is smaller than that coming in because it's getting absorbed. And so you make a measurement without the sample, make a measurement with the sample. You know how much light was absorbed by the sample. Then you take the ratio, therefore you know how much absorbance it is. Change the wavelength, do the same thing. To make a plot of all different wavelengths, and that's how this process works. The essence of spectroscopy. So if you have a single chemical, the people have been doing this for a long time, and it actually works remarkably well. And this is in fact non-destructive because you are just sending in light in, right? So there are other ways, mass spectrometry or some other. There are several different ways of doing this kind of figuring out what molecule it is. But this one has the advantage that it is non-destructive. It's like just you're sending light in, they're not destroying it in any way. After that, if the sample is still there, you can do some more analysis, you can do chemical analysis, something else with it to do some kind of figure out what else is going on. Okay, so what's the point of finding the chemical composition? Say in some diseases, the chemical composition could change, right? So if you look at measure change in chemical composition, you can figure out what disease it is. So we'll get there in a minute, but like that's the reason why biologists and my medical, like for medical purposes, clinical purposes, why this could be useful. So if, as I mentioned, for one chemical, you can figure out what's going on. But what if it's not one for one single uh, chemical? If it's a mixture of chemicals, for example, this is a typical spectrum of a tissue that you would get. So tissue is like something from the human body, um, a part of the human body. So there are all these different small peaks and big peaks and different at different wavelengths and wave numbers. A range of things happen at all different wavelengths. So this is because, as you can imagine, yeah, the body is not just made of one or few molecules; it's a bunch of molecules put together. So even then, like so, because it's a complex mixture of molecules, so what can spectroscopy tell us? It can still tell us, say, the big category of molecules. For example, is it a lipid? So lipids have some peaks in the few, some here and some over here. Then you have proteins which have like peaks in some of these regions, and then you have nucleic acids which have a few other peaks over here. And carbohydrates. So all the different glucose and like fructose and other carbohydrates, in which when you like take it inside the body, they go into different parts of the body, right? So basically, you can figure out the gross molecular composition of tissue by looking at spectra of this sort. Okay, so from this kind of a spectrum, you can figure out so like whenever there are changes. So to give you an example, say the nucleic acid, it's there in the DNA and RNA, right? So therefore, if the amount of DNA present in a particular cell, if you, I don't know if you have, any of you have taken classes in biology, one of the, when cells, so one of the basic things that you learn in, like one of the first classes in biology is cell division happens. Like one cell becomes two cells. And what happens is the DNA makes a copy of itself and it becomes two, like there are copies in each cell. And that's normally normal cell division. But when you have cancer, what happens is that cells divide very rapidly. So there are a lot of this cell division happening. The amount of DNA becomes much more. So if you can measure the quantity of nucleic acids inside in a region of, in an area, if there is cancer, it's possible that the amount of nucleic acids is increasing much more than the other proteins and other things. And therefore, it tells you something about, by measuring the chemical composition, it tells you if there is a disease, if there is cancer growing or something of that sort. So that's like that's of course not all, like sort of, this is like one of the many things that happens in cancer. So this is one of the measures of one. So this is one data point, is one measurement which tells you something important about cancer or not. There are other like 20 different other things which happen, which can be measured through the spectroscopy, and you can, by looking at the changes in this chemical composition, you can figure out whether a disease is there or not. So that's the essential idea of these class of approaches as it applies to biomedical applications. Yes. Mm-hmm. A few peaks. Yeah. Not one peak, but multiple peaks, a combination of peaks. Yeah. Each bond will have a one or few combination of peaks, and you have multiple bonds, therefore it will be a combination of multiple peaks. And the amplitude can be different. Yeah. Mm-hmm. 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 That's a good question. So right now we are doing it only for what's called biopsies. You actually take a needle, get to the place where you suspect. So you like, so this is right now the in fact that's the reason why it's an active area of research. People are trying to figure out what you're saying. It's not yet there. It's like sort of we are in the literally in the middle of developing the methods to do that. It's like there are so many things that yet not yet discovered. So one of the things is you have to put a needle and get, like if you're suspected, say, of breast cancer, right? You have to put a needle, go to the location where there is a lump or something of that sort, take a sample, and then in and put it under this instrument and do the measurement. So it is like in the future, we hope that we will get to what you're saying. But as of now, it's it's in the like stages of development where you have to actually look at it after you take it out. Okay, that's one of the things which people are working on. That's a good question. Any other questions? So I want to talk about how this instrument works, like some of the details of how exactly this kind of instrument, this type of instrument works. So the first, like there are several generations of these instruments. So the first type of instrument which did this was what's called FTIR, Fourier transform infrared spectroscopy. Okay, so that is one type of instrument which I'll describe, and then I'll talk about a new kind of new method called photothermal imaging, which is like the latest one, latest method to make measurements. The previous method, and so you basically, in order to get a spectrum, what you do is like have a detector where like sort of if light. Have a light source, a mid-infrared light source, which emits light and falls on a beam splitter, 50-50 beam splitter. One goes to one mirror and another goes to another mirror, and it comes back. So these are both mirrors which reflect light back, and then they get recombined and goes to a detector. Okay, so this is the essential. So you put a sample in between here, but for now, let's say you just have a detector over here. So the idea is what happens at the detector when the mirrors are basically perfectly matched, right? So there's constructive interference. Wave from one and wave from the other come back. They constructively interfere. You get a high signal at the detector. So M1 plus M2 is the overall signal, which is as you keep one mirror fixed, keep moving the other, and as a function of that mirror, you can get like this particular wavelength, they get amplified. But then, if there is a phase shift, if there is a kind of if the mirror is moved a little bit, then there is partial interference between the two. You get a signal of this sort. But if the light from one mirror and the other mirror, if the mirror has moved enough so that they basically the maximum of this and the minimum of this meet each other, then they destructively interfere. So basically, by moving the mirror and making a measurement at the detector, you get kind of a signal of this sort. This is called an interferogram. And so once you get this, and this is like by moving this one mirror and making a measurement, you can make. So you keep making these measurements, and so once you make this set of measurements, you take a Fourier transform of this particular data, and you get the spectrum. So what we're measuring, so keeping one fixed and the other moving. But there's some feedback. Yeah? It's okay. All right. So by keeping one mirror fixed and the other moving, you are essentially measuring the autocorrelation function of the source. That's the essential idea of being used. And from the Wiener-Khinchin-Einstein theorem from yesterday, so like you can, by taking a Fourier transform, you'll get the spectrum of the source of light that you're measuring. So that's the essential idea behind this approach. And so this is therefore because you take a Fourier transform, it's called Fourier transform infrared spectroscopy. You're measuring something as a function of wavelength or wave number. It's infrared light that you're measuring, and the instrument uses a Fourier transform as the crucial way in which you can get the spectrum. That's the FTIR. So this is how you can make measurements at any given point. At one point, you can get this, like for one chemical, for one location, you can get a spectrum of this sort. Then the next question is, if tissue, like if you have one chemical, you just make like it's a powder or something, you just make a measurement and or liquid or powder, it's easy to make, it's a homogeneous thing. So it's not varying from place to place. But if you have tissue or some biological sample, from one position to the next position to the next position, there is a big variation in the spectrum, right? So one point you have a spectrum, another point you have a slightly different spectrum, and another point, because the chemical composition at every position is different. So when you have chemically heterogeneous samples, meaning when you have different locations have different molecular composition, then you can't just do one spectroscopy, one measurement, which is just spectroscopy. You do what's called spectroscopic imaging. You combine spectroscopy with microscopy or imaging. So you make a spectral measurement at one pixel, a spectral measurement at another pixel, and so on. So you get an image along with spectrum in spectral information at every pixel. So that is what's called spectroscopic imaging. So this approach is called mid-infrared spectroscopic imaging. So that's the big technique that we will people use to my lab and other people use for these kind of measurements. So let me first talk about the FTIR imaging system. So the previous one was FTIR spectroscopy, just a single spectral measurement. Combine that with spatial information. How do you kind of get imaging information? That's the next thing I'll talk about. And then I'll talk about some of the new methods, new completely different ways of measuring without using this Fourier approach. There are other approaches to making measurements, which I'll get to in the second half of today's lecture. So FTIR system, it looks something of this sort. It's a cartoon schematic where you have a source of light. Light comes out and goes to a beam splitter, goes to a mirror, comes back, goes to a second mirror, comes back, and then it goes to the sample. So the important thing is, instead of light just going through the sample, it's focused using a pair of lenses onto a sample. And so our lenses are reflective optics, Cassegrains. It's focused onto a location in the sample, and then light comes out and is focused onto a detector. And the detector could be one point detector or array detector. It could be a camera sensor type of a big detector. So you can get like an image instead of one spectrum alone. And you move one mirror, keep one mirror fixed, move the other mirror. At every mirror position, you collect detector data, a camera image or a detector data point for every position of the moving mirror. Like you collect data. That's the idea. Okay, so here is, so this is a cartoon of how this works. So you're moving one mirror, and you're fixing that, and then you're moving the sample on a stage, and you're getting data over here. So there's movement here, and then this, the sample itself is moving over here. XY movement, and every image you get this data, and what you get at the end of it is something of this sort. You get a cube of data. So this image is just like a microscope image. It's like just a slice image of one, like say at one wavelength you get an image, but at every pixel on this image, on the third dimension, the Z dimension, you get a spectrum. So it's a data cube instead of a single. It's not a picture, it's a cube of data that you get. And this axis is the wavelength or the wave number axis, and like just XY axis is, so this is the X. If you think of this as the XY axis, XY is just the image, Z is the spectral dimension. So X, Y, Z. So 3 dimensions is a cube, data cube. And you can measure in the infrared, you can measure up to even 1600 wavelengths. So imagine in like visible light, right? So you have red, green, and blue, three colors. Typically, that's what you get in most color images. See, red, green, blue are enough to kind of get images in the visible. But in the infrared, because there are so many, such large number of wavelengths, you can get up to 1,600 separate wavelengths, and in FTIR, they're commonly measured. You may not need all of them. It's like depending on the molecular composition of what you're looking at. You may need much fewer number of measurements, but like that's the difference in compared to visible light. When you use infrared light, you have a lot more information about molecular composition at different wavelengths. That's one of the advantages of infrared of this sort. Okay. And once you at every pixel, you collect the spectrum of this or the tissue spectrum that I showed you. And the basic idea, the basic goal of this approach is instead of we look at the chemical at every pixel, the chemical, you can have a guess of the estimate of the chemical composition. So one pixel, chemical composition, another pixel, chemical composition. So at every pixel, if you know the chemical composition, by looking at the chemical variation in chemical composition, you can figure out whether there is a disease or not at a particular region of tissue. So that's the essential idea of this type of approach. Okay, so you can do data analysis, and figure out what type of tissue it is, whether it's cancer, if it's cancer, what grade of cancer, and so on and so forth. So people used to literally look at the spectra that you get, and I try to figure out what chemical there is. But when you have a mixture of chemicals and you have such large data sets, like this image could be like a 1,000 pixels by 1,000 pixels, right? So a million pixels and million spectra. It's simply not possible to look at every spectrum and do an analysis. So you need like big data methods. Out of necessity, you need methods which kind of computational methods which take the data and do analysis to give you disease like diagnostic information. So what the kind of approach with my previous lab and several others have taken is use machine learning of different sorts. Okay, because this is a big data problem. So what you do is start with the data, do some data reduction. So kind of reduce the amount of data through some data reduction methods, while keeping all the important information. Then use different kinds of machine learning methods. So, for example, if you want to do prostate cancer analysis, which was some of the thing that I did in PhD. So, prostate cancer, there are 10 different tissue subtypes in tissue. So, you do what's called supervised learning. So, machine learning, there are two broad categories: supervised and unsupervised. Supervised means you know something about, already have some prior information. This region is this class, this region is this class, and so on. Unsupervised means you don't know all those things. You kind of cluster or do something to kind of categorize it naturally. But in supervised learning, you already know some class information and you train the algorithm to to identify the specific classes that you already know. Okay, so the idea here is that you start with so in fact we have been doing this since 2006, 7, 8 time frame. So it's long before machine learning became a popular thing in the real world. But so we started Bayesian algorithms, genetic algorithms, neural networks, random forests, convolutional neural networks, more recently deep learning. Most of these algorithms, you probably have heard of, we have tried them, and it works. Turns out that all of them work remarkably well. Okay, so each one has some advantages and disadvantages depending on the specifics of a particular problem. But in fact, I wrote a chapter on machine learning in 2010, and one of the kind of the things that you learn if you learn machine learning is that there is something called a no free lunch theorem, which essentially says the exact algorithm that you use may not matter too much. it matters only to the extent that there is something specific in the data which allows you to exploit that, use that algorithm. And any one algorithm will have an advantage compared to another only for us, like sort of because there is something in the structure of the data which makes it useful. Okay, so it's kind of the exact algorithm like may not be relevant if unless there is something specific in the data which makes it useful. Okay, so but in any case, you optimize it after compared to histology. You have doctors who mark regions out and like different subtypes and so on. And we work with doctors who do this. And like after optimization, we get analysis and results where it turns out that for breast cancer and prostate cancer, which was some of the things that we worked on, and for like and this work has been continued in my previous lab, and like they have extended this to more than thousand prostate cancer and thousand breast cancer patients and so on. So it has been validated quite extensively, and you get like 0.99, 99% or so accuracy for tissue subtyping. So it's for say the prostate cancer, identifying 10 different cell subtypes. Okay, so that kind of for that kind of an analysis, it works remarkably well. Okay, so I think this is a good time to pause. Yes, question? Yeah. Unsupervised learning, yeah. Sure. Yes, so this uses supervised learning, yes. What I'm showing here is supervised, but there are, yeah, sure. I can tell you about unsupervised. So unsupervised learning uses the fact that, like, sort of, if there are some natural groupings in the data, let's say, after you get, say, it's two, right? Let me give you an example, okay? So let's say there are measurement, like you make a two-dimensional measurement of some sort, right? Like along the x, like say x1 and x2 are the variables, right? Data point one is here, data two is here, data three, four, five, so on, like this, right? And not like when you look at the data, like the data naturally has one cluster here and another cluster here without even knowing anything about it. If you use what's called, for example, an algorithm for unsupervised learning is what's called k-means clustering. Okay, k uh, k-means, yeah, clustering. So it naturally, like, and if you say there are two clusters, find me the clusters. Then it will kind of figure out that this is one cluster, this is another cluster. Just naturally, the data is spread out in such a way that there are groups, and that's if it so happens that your data has this feature, then this approach works. But quite often the groups that it comes up with may not be what you want to, like it may not correspond to exactly the, say, cancer or some, it could be something else, right? For example, it could cluster like if you have data of different types, it could cluster men and women. Although that's useful, it doesn't say anything about cancer, not cancer. The clusters it comes up with is maybe irrelevant for what you're working on. So whatever the natural clusters are, those are like unsupervised learning methods pick up on that. Okay, does it answer your question? Yeah, sure. What's your question? Yeah. So there are several different unsupervised methods. So hierarchical clustering is another one. Key means is another one. So there are different ways in which you can do this analysis using like building on principal component analysis is another way of approaching this problem. So there are several different ways in which you can kind of if you don't know much about the data you're looking at, if you are like it's kind of you don't have prior information, then unsupervised methods are the way to go, where you look for natural kind of variations. If you know, like, okay, these are the 10 different types of cells that are present or tissue types that are present, That information is very valuable, and then you can use a neural network or random forest or like different methods, genetic algorithms, to basically extract information and separate it out into your what you already know is the kind of class. Okay. Is there any difference? Say that again. Reference book, yes. Like I wrote a